Thanks, Taryn. Hello, everyone. Thanks for the introduction. I'll be talking about linking thermal tolerance and telemetry, and then on mullets. So I'm just trying to figure out which one goes where. Okay. Okay, so the first time when I started this project, which is my PhD, I was looking for pictures to present, to add to my presentation on proposal. And I said mullet on Google, and I went to photos, and this is the first picture that came. So I just wanted to just take a moment and appreciate that. But we're not talking about this mullet. We'll be talking about the humble mullet that I think everybody in this group should know it. Because when you even search on the internet and look at what species can be caught using a mullet, usually the answers you get is like almost everything can be caught on mullet. The right question to ask is what can be caught on mullet. So, yeah. So, obviously mullets are being caught a lot because they are a prey species. So I'm talking about generally mullets, but then I'll be focusing on a certain species of mullets. But the reason for this is because knowing where prey species are can feed into models about finding where the predators are. So I think the predators are more the focus for the group, but yeah. So mullets and other prey species, their distribution is dynamic. It's not in the same place, so they are controlled by environmental factors. And then temperature has been identified to be one of the key abiotic constraint to the movement of certain fish species. So the question here is what mechanism link temperature to movement? And then I'll be using flathead as an example to try and answer that question. The reason flathead was chosen as an example here is because of its global distribution and being at the base of the food web, just like most mullets, and also it dominates a lot of fish assemblages in the coastline of South Africa. Okay, just to get started, so we used critical thermal methodology to look at the thermal tolerance of mullets, and then I think this has been explained a lot over this week, so I'll just touch quickly through the methods. So we had a lab set up at Rhodes University, and then where we introduced the mullets into the system, and then we rammed the temperature up and down to see how they react to that. And then the results of our experiments, the CTmin on the other side and CTmax with individuals on your y-axis. So there was variability, but not too much in the CTmin and the CTmax, with some individual being more tolerant to higher temperature compared to lower temperatures. But the general pattern is that mullets are highly tolerant to a big range of temperatures, which is just the big takeaway from this. And then we continue to look at the orbicular beat movement of mullets in order to determine when do they start experiencing physiological stress. And then with that, during the same experiments, we had cameras in the water tanks to see the orbicular beats, which we counted and plotted here. And then we modeled to see the breaking points, which are these dashed lines here. And then we see that the mullets start showing physiological stress way earlier than their limit. So at 13 degrees and 27 degrees that are shown there. So the next question from there was that do these fish get to experience these temperatures in the wild? And then we plotted the temperature of the wild. Yeah, so this fish were collected from the Kowie Estuary. So we collected temperature data from the Kowie Estuary over a 1-year period. And then there's the distance from the mouth of the estuary going all the way to the river part of the estuary. And then as you can see with the dashed line, I was going to use a pointer, but I don't know where it is. So with the dashed line here is the breaking point that were shown by the mullets. So you can see that it overlaps with the temperatures that they do experience, although not all the time, so it could be like a seasonal breakpoint limit. So around June, which is winter here, and in summer not much, but close. Okay. So with that information, then we also thought, let's actually find out more by taking fish in the same estuary and see if this information will match what we see in the experiment. So we went to the Kowie, and I think by now everyone knows where Port Alfred is. So we went to the Kowie and put some receivers in the water and also some temperature loggers. And then we tagged some fish. We caught some fish, tagged them in the Kowie, and then monitored them for a period of 1 year. And this is where the fish were tagged. Okay. Yeah. So this just shows general movement of the fish, individual fish over time. And then if you look at it, you could think that the fish are generally using the entire estuary over the course of the year. But then when we actually divided this into seasons, yes, they do use the whole estuary, but there is a proportion amount of time that changes according to season as to where they are in the estuary. So in the lower reaches, they don't spend much time in it, but the middle reaches, that's where they spend the most time. And then the upper reaches, they do spend time, but then what was interesting was to see that in winter, like here, they spend 1%, which was the lowest time that they could spend in the upper reaches compared to when they go all the way to the middle reaches, and there's less variability in how much time they spend in the middle reaches in winter. So that was an interesting finding for us. And then we also looked at other environmental variables that could drive this. Obviously, sea temperature and river temperature came at the top, and then we also realized that river flow also had an impact, but rainfall did not. So, just to bring this back to the whole reason we wanted to do this and to look at this, was to look at in winter and summer, like how are mullets affected by temperatures in the Kowie Estuary. So in winter, we know that cold riverine water temperatures drive the mullets out of the upper reaches all the way down into the middle reaches. And then we know that in summer, there's still warm water that could also drive them down. But then in South Africa, we have this cold upwelling seasons that come in summer seasons. Cold upwelling that happens in summer season where water can reach up to 8 degrees. So potentially on days where there could be cold water, it could also push mullets in summer season, could push them all the way up into the middle. So mullets could be actually being squeezed in the middle during the summer season. And then climate change, as we know, we know that the impacts of climate change going forward is the increase in intensity and frequency of this kind of upwelling events, which could put mullets also in a difficult position. And then to answer that question that I asked is what links temperature and movement, we think that behavioral thermoregulation could be the linkage between the two. Of course, for physiology, we did not look into other aspects of physiology, which could be the next steps for us instead of just looking at temperature. But then temperature so far was shown as a very clear driver of this species movement, because it can drive the fish to find a refuge. It can drive them to look for a refuge in the system, which means that they could be limited in where they occur during certain seasons. And that may affect some predators that feed on them. Yeah, like the larvae that also feed primarily on mullets. But yeah, so implications for this include the climate change. We are concerned about the mullets with the impacts of climate change showing right now and into the future, and then which means that they could climate change could compress the prey availability, spatially or temporary, in the system, and obviously that will have an impact on the predators that feed on them. Terran did a nice study on the leofis movement in the same system 10 years ago, and their movement was also shown to be affected by temperature, but it will be interesting to try and look at predators and prey around the same time and see if their movement will follow each other or not. Yeah. So in conclusion, just trying to say from the study that obviously environmental factors are affecting prey movement, a lot of fish, by affecting their physiology or aspects of their physiology, and which can also affect the habitat that is accessible to them that they can use, which in turn may affect the distribution of predators and potentially recreational fisheries success in the future. So I would like to thank everyone that was involved in making this project possible. I tried to squeeze it in very short time, so I feel like there could be more that we could talk about over lunch, but yeah, so thank you very much for listening and for the funding to attend this conference. That's a very excellent question that I also was thinking about it when I said we need to now tag them at the same time, the predators and the prey, and see what's happening there, because Terran studies show that they might be also be driven by temperature, but I don't know how much is limiting the leofis from maybe using the upper part of the estuary versus using the lower part. But we know that for mullets, they're limited by temperature, so we could potentially find out what could be driving both. Yeah. No, there's no need.